welcome, or welcome back. And yeah, so we're back for the biochemistry lecture. We missed last week due to, a very slight misunderstanding. But as mentioned in the Discord, we should be back to our regularly weekly intended biochemistry lecture. So don't want to waste any time. Gonna try and get right into it. One moment. We're just gonna wait. But yeah, apologies for last week. But yeah, these things happen. And yeah, the intention is weekly lectures. And well, hopefully long term, the ultimate hope is like to cover an entire textbook of biochemistry material. Should be starting any moment. Just waiting on TALORX to hop in the channel. One moment.
Hello?
Yeah, can you hear me?
I can. Hello. Let me get my camera on.
Yeah, I'm gonna do the same thing.
I'm gonna close this just in case my partner comes by. Yo.
One
There's-
second.
Yeah, all good. There's these clips that go on your door so that only a cat can get in and it, like half closes it. They're amazing. That's
It
what's
sounds amazing.
on the door.
Second. Do. Oh, thankfully, even though I updated my OBS, it seems to have kept my settings for now.
It didn't fuck everything up?
Not yet.
Good stuff. Why? There we go. Okay, let's remove guest video and then re-add it. Sorry, everybody. Just uno momento. We're just getting everything sorted here.
Indeed.
I'm looking for the word window. That's what I'm looking for. There we go. I found it.
I have a window capture, and yet it's not capturing my window. Well, first I'll start sharing my screen with you.
Sounds
You
good.
can see that.
And let's see if... Oh, that doesn't work.
Alright, hopefully you're able to see that.
Okay, yes.
Well, I have the
Watch.
red curtain up.
Amazing. It looks beautiful. Let me fucking make this full screen. Actually, if I full screen it, yeah.
I don't want to give things away.
All right, I always feel bad because I do your camera such a disservice. Because your camera's great and it looks really good coming through my Discord, but because I have to blow it up so much, it makes it not look as good. But it still works.
Whatever works at this point.
Yeah, it still looks decent. I'll make myself a little smaller. Perfect.
1 second. I think I might be sharing the wrong well, it doesn't really matter at this point. As long as you're seeing stuff.
Yeah. Yeah. I'm gonna crop out the bottom and just go to the... Okay.
Oh, whoopsies. I think I messed some stuff up on my side. 1 second.
Okay.
I resized some stuff.
All good.
Okay, I'm not going to mess with that anymore. Oh, I know what I'm doing wrong. Actually, 1 second. Well, we'll just go with it for now. I don't want to jinx what I had. Cause yeah, I think on your end, you're seeing like a timer and stuff, right?
Yeah, like...
Yeah. I don't want you to be seeing that.
Yeah, I feel like I'm in the presenter mode. It's in your presenter window.
Yeah.
If you go to your window capture, like, it looks like there might be a different window.
Yeah, no.
Do you have a different screen?
Yeah, I'm going to rectify that. I just dragged out the wrong window.
Nice. I'll let you know if it changes on the side here.
Yeah.
I guess you can probably see it too.
Okay. That one should be a little bit better. You shouldn't be seeing a timer. You should mostly seeing just like a browser.
It doesn't.
Oh no, not
changed
sorry.
on my side.
Yeah, sorry, I didn't change on your side. I changed on my side. Still a rookie. Not this. Yeah, okay. That. Now it should be better. Or you're just seeing a browser. So the question is, can I fix it on my end?
The curtain is giving me suspense. It is very suspenseful. What could it be? What could it be?
But now you are seeing a different window, right?
Yes, yes. This seems like the correct one. Like, yeah.
Yeah, I need to make
It's actually the presentation. Or the other one is like the presenter.
Yeah. Okay, I think I've got everything.
All right, let's go. I'm stoked.
Let's see. One second, just doing some last minute checks on my end.
Get my window on the screen here.
Okay, good enough for now. All right, I think, well, we're gonna go with it, and hope for the best.
All right.
All right, so everything's set on your end. You can hear me?
Yes,
Everything?
I can hear you. It sounds like chat can hear. It seems like it should be all good on this side.
All right, apologies for the delay. You know, still
All
ironing
good.
out the kinks.
All good.
All right, you
I
ready?
apologize for the delay in classes by missing last week. So
okay.
fucking
Not a problem at
I
all.
apologize.
You get 3 absences before you're kicked out of, you know, the
Really?
university.
Holy shit, that's a lot. God damn. I plan on not using any more.
No, you totally can. You ready for the drum roll?
Yes. Audio's
All
on,
right.
we should hear it. Oh shit! I hear it this time. I think chat should be able to hear it.
All right, we're starting out since we missed last. We're not. Well, we are reviewing basically since we missed a week. I want to make sure that we pick up where we left off, and so we're starting with a question that we asked before technically. In terms of in the structure of DNA, what makes up the backbone? Okay, and I obviously can't operate my slides. Is it A, cartilage? B, adenine and thymine? C, nucleotides? Or D, deoxyribose and phosphate?
The answer is.
Oh, I can do a
I
drum roll.
believe. Oh, you can do a drum roll? Oh shit, let's go. I believe it's D, final answer. Deoxys and fucking phosphates.
Yep.
Let's fucking go.
You are correct. All right, so information retained, fantastic. Although there's still more, so we're gonna do a little bit of a quick review of
Yes.
DNA,
I'm
and then I
down.
think today we'll have sort of at least 2 things on the agenda or on the course outline for today. Is we're actually gonna briefly cover some research, some brand new research, like a couple weeks old, that has to do with what we covered, just a couple weeks ago. Do you remember Luca? Does that sound familiar?
The last universal common ancestor.
Exactly. And conveniently, just in the past couple weeks, there has been research published about Luca. We're going to get to it right now.
Shit.
Okay. Where, as I highlight there, I don't know if hopefully you can see it. Might be a little bit too small.
I can see it.
This was published July 12th, 2024. So like 2.5 weeks ago. And the title of the paper is The Nature of the Last Universal Common Ancestor and Its Impact on the Early Earth System. So if you recall, it's okay if you don't, but when we went through Luca, and like where life began, and I think there was even a question, approximately when did we put like
350 million years.
is that going to be your final answer?
It's in the 300 million years range, either 350 or 320 or something like that. 350.
So you definitely have 3 like you're like, I understand the 3, but you might be off by, instead of million.
Oh, sorry, I'm dumb. Billion.
No, you're not dumb. Yeah, where pretty much that's the currently, accepted, date of when life began, or when Luca.
3.5 billion.
Exactly, 3.5 billion years ago. But as highlighted here, this is still an ongoing, active area of research of like, is it really? Like, multiple lines of evidence point to that. This is not my strict area of expertise, but this is what's taught in the textbook. Like the figures that I've showed you, the straight pulled from the textbook that I essentially learned from. But we still don't know most of the stuff about Luca. And that's what this research was about. It's like doing a little bit of a reevaluation, okay? And so I'm just gonna go through some of the brief highlights, where this is just like the introduction. So like from the introduction summarizing why we currently think all cellular life evolved from one single cell. They have a nice little paragraph. All papers that start off, they have a nice little introduction giving the background. So as they say, the common ancestry of all extant cellular life is evidenced by the universal genetic code, which we have seen DNA, and all cellular life has, DNA in it, okay? And it seems to be universal in its structure and in its code. The machinery for protein synthesis, which we'll get to at some point. I know some technical jargon of shared chirality of the almost universal set of 20 amino acids. So we're not going to cover chirality because it's not super important yet, but we're going to be covering amino acids today, as in that's going to be the start of proteins and amino acids. So we're going to cover that or introduce that today. And then they're saying the use of ATP as a common energy currency. So what they're saying is like, based off of all these things that we find in cellular life, okay, this is why we think like it pretty much all originated from Luca, and we have all of these things in common. We all use the same building blocks, the same 20 amino acids. We all have pretty much the same protein synthesis machinery. We all share the exact same DNA architecture. As I mentioned before, if I take some, human genetic information and I put it in bacteria, okay, the vast majority of the time, like the DNA can like use it, or can use it to some degree. And then, the last universal common ancestor is the node on the tree of life from which the fundamental prokaryotic domains, so we covered those domains, archaea and bacteria, diverge. Remember the branches on the tree of life of Luca's at the root, or that's how we view where Luca is. And then as such, our understanding of Luca impacts our understanding of the early evolution of life on Earth. Was Luca a simple or complex organism? And that's essentially what this paper was about, or trying to, get more information about Luca. Paint in a little bit more of the picture. We're not gonna do a deep dive. We're just gonna go over some of the highlights. But actually, after the lecture, I'm gonna cover the paper in more detail. On my own stream. But you don't need to understand this, okay? But as I say here, this will all be understandable in time. What this figure is this is how we depict metabolism, okay? All the pathways, okay? All the biochemistry pathways that are occurring in an organism. And in black there, okay, that's what they think the metabolism, okay, all the chemical reactions that took place in Luca based off of their research, okay? Where you can see that there's a lot, it almost looks like circuitry, or as we're going to see, there's a lot that goes into metabolism, okay? You don't need to like really know this, I just want to introduce it and just highlight also how complex it is.
Yeah, that's cool.
There's a bunch of stuff there, in the background, and in black there, it's like, well, this is what we think the metabolism of Luca looked like based off of the research that they did in this paper, okay? And then in another figure in the paper, so they have their own version of their tree of life, okay?
Lika!
So
what is Lika? Sorry, you don't have to answer that.
No, it's good. No, it is. It's totally related. I put it there.
Lika Labaka?
Yeah. So here's where so you can see Luca's at the bottom, right? And then it goes LBCA, Labaka, Laka, and Leka. So their nomenclature. So if you remember, okay, the three domains, okay, eukaryotes, prokaryotes, and archaea. So
That makes sense.
Luca's the universal common ancestor. BCA is the bacterial common
Yeah.
ancestor.
Yeah.
Laka is archaeal common ancestor, and Leka, that's where we come in. We're the last eukaryotic common ancestor. So that's where they're putting, their branches. Okay,
Yeah.
and you can see below Luca, okay, there's something below it. Obviously, it's this undefined gray area, and they even see
Is that new? Is that
uh,
gray area the new part?
not fully. Well, so basically, it's like the current sort of understanding, but again, this is not my area of expertise. But Luca's like that's where he put that's where cellular life began. But there was something before cellular. Right? It didn't just go. Yeah, exactly. Where it's maybe some mixture of like chemical stuff, okay, and then somehow cellular life formed. And so there's something before Luca where it did not just go from no cell to cell. There's some sort of like intermediate there.
So did the gray ones die off, or are they still in existence?
From what we can tell, the gray ones, like we have basically, if they're still around, we don't know it, and we have no, at least currently, we have no tools. We have no tools to see if they're still around, or they just no longer exist. Where
they could still be around, and we don't even know it.
Exactly.
The gray ones.
And then the other figure, okay, that's their representation of their Luca looked like based off of their research. You don't need to understand it. Just like you think about it, it's like that's their version of the first cell. Luca looked like this. It had this chemical stuff going on. It had these, amino acids, and as shown there, it even had an early immune system. Okay, and well, as we'll see at some point, everyone now knows about CRISPR and Cas and all that stuff.
Yeah.
Well, CRISPR is actually, we have sort of, at the end of the day, its original function, it's from bacteria, and it's part of the bacterial immune system, okay? And we as humans, we have co-opted it for our own genetic engineering purposes. But we call it the CRISPR-Cas system, okay? And this Cas system, that's pretty much, so it sort of like begs the question of like, well, if you're the first cellular life, then why do you need an immune system? What are you defending against, right?
Oh shit.
But and that where what they show in the paper, and again, we're not going to go into detail, just going to cover one more thing, the conclusions. But based off of the research, they essentially, their hypothesis or theory, it's like, well, Luca evolved like already in an ecosystem, as in like already stuff was happening. It was just not like, this one single cell, evolved into existence, and then it's like all alone, nothing around it. That's where like that gray stuff, on the tree of life that you were asking about, which is a great question. That's like, yeah, it seems like it, came into existence, in the context of like something where there.
It didn't fall out of a coconut tree. It came into existence within the context.
Just same thing with humans. We don't fall out of a coconut tree, okay? We are all brought into a system, an ecosystem, and no person is an island. Even Luca did not appear to be an island.
That shit's deep. That shit's fucking deep.
Exactly. So, wrapping up, just briefly highlighting this paper is, their conclusions, okay? The nature of Luca's metabolism has proven equally contentious, okay, because it's hard to go back, 3.5 billion years. And so, based off of their research, okay, they are putting it more back, as in, instead of 3.5 billion years ago, and I showed you
it was 420 billion years ago? Let's fucking go!
4.2 billion, so 700 million more.
4.200000000.
Exactly. And this is just based off of their research, which appeared to be, lots of researchers were involved in this. This was published in a very respected journal, and it's also been in headline, certain science headline news. But we're all I really want to highlight is we're always revising, looking back, making sure that we're getting things right, maybe at some point, okay, the 11th edition of the textbook that we're covering will have 4.2 or 4.0 billion years ago as Luca. But still, the currently accepted, in the textbooks, it's still 3.5 billion. But just to show that scientists are still trying to understand and pinpoint down Luca and what it looks like.
That's amazing.
And then fully wrapping up their conclusions just in the bottom there, where another interesting factoid, where if the Earth is 4.5 billion years old, and we're now putting Luca at 4.2 billion, basically, it was about 300 million years before the Earth began and, like, the first life form, And so that's not actually a lot of time, at least when it comes to evolutionary history, right? Where it only took, it's pretty much like almost somewhat instantaneously did, Earth come in, did Earth form, and then a life somehow took off, or cellular life took off. And even then, even before cellular life, there was, something before it. But yeah, as they say,
cellular life.
Exactly. How evolution proceeded from the origin of life to early communities at the time of Luca remains an open question. But the inferred age of Luca, 4.2 billion years ago, compared with the origin of the Earth and Moon, suggests that the process required a surprisingly short interval of geologic time.
God damn.
Short window. If in fact this is true, but this is still, this is not like, stamped of like, oh, official approval, let's put in the textbooks tomorrow. But it seems to be pointing in that direction. So yeah, just wanted to highlight that. And then now getting into whatever.
That's a huge jump, dog.
Yeah,
fucking
exactly.
Even like from like 3 to 4, like that's a big jump. Like fucking, like I guess like 3.5 to like 4.2, like almost a billion years is like a pretty gnarly fucking pushback. Like that's not like, oh no, it's not 3.5, it's 3.7, like that's big.
Yeah, it is big. Like they're pretty much, 25, about a quarter, quarter, 25%, like whatever off, if in fact true. And just shows the iterative process of science.
So are they like very, and I know it's a very different field, so that's why I'm like thinking that they're probably like beyond confident through a myriad of different sciences or fields of science. Is there any possibility of pushing back the Earth's age at all? Or is it like, no, that's fucking confirmed by a billion different things, like that's solid?
Yeah, there's let's say more lines of evidence, and not to downplay any sort of scientific research, but that's a little bit easier, so to speak.
Yeah. Like it's a different question for sure. Like it's able to be checked by more things,
Exactly, because how Earth started and Earth today, lots of changes occurred, but still, like, there's still evidence of early Earth, as opposed to, like, early organisms. Organisms go extinct, wiped from existence, and not only do, like, they have to be in the fossil record, but we also have to, like, discover them as well. Just because there's a fossil in existence doesn't mean that we actually know where it is or that we can find it. And that's one of the challenging, at least from what I can tell, based off of me reading the paper. It's like, one of the challenges, it's like, sort of built into the technique. So I'm going to go over the paper in more detail, after the lecture. But their technique is, they're trying to go back in time in evolutionary history,
Yeah.
And how they do that, part of that is through, DNA analysis or genetic analysis, and tracking genetic changes over time. And there's ways to do that. But ultimately, they're looking at evolution and evolutionary relationships, But if that's what you're doing, then there becomes a problem in that, if your technique is analysis through evolutionary relationships, then what do you do at time zero of, like, no evolution has occurred? Evolution hasn't started yet, Sort of like thinking loosely of, like, if you want to observe a black hole, like, what do you do when you shine light at a black hole? From what I understand, no light escapes a black hole. So how do you directly observe
How do you do it?
It's sort of like a similar thing of, like, if you're trying to look even before the first cellular life, like when there's no evolution, and you're always taking an evolutionary approach, like you're sort of like you're going to hit a wall at some point that becomes almost impossible to overcome, or you have
Yeah.
to find some other way. But yeah, all evidence seems to point there was something before Luca. We don't know what it is, and it doesn't seem like as of right now like we can like access it or have any idea of like what it really looked like. But all areas of active research, I'm sure at one point they'll figure it out. But yeah, just wanted to highlight that and show that, you know, the scientists are working on it. That we're still interested in Luca. So picking up where we left off, okay. We covered some DNA last time, and now we're gonna start off pretty much, diving back into DNA, just giving a brief review on it, what we covered last time, but diving into, you know, questions, okay? As in, do you now know the fundamentals of DNA? But if you don't, we're gonna recover DNA. So
let's go. I think I got it.
Nucleotides are composed of 3 components. What are they? A, adenine, thymine, and guanine. B, cytosine, guanine, and adenine. C, pentose sugar, phosphate, and nitrogenous base. Or D, hexose sugar, phosphate, and nitrogenous base.
Oh, you got me with the pentose or the hexose. I'm pretty sure it is pentose. My final answer is C.
C. All right, we're going with the drum roll.
Drum roll, please.
And that should be correct.
Yes, let's go. Hell yeah, let's go.
Pentose sugar, phosphate, and nitrogenous base. But we're not done. Okay.
Let's go. I love this question.
When cytosine and guanine base pair, how many hydrogen bonds are formed? Is it A, 0, B, 1, C, 2, or D, 3?
Okay. Well, my answer is 0. Because like it's not a fucking hydrogen bond. They are attracted to each other. I know C and G and T and A go together, I believe.
You absolutely nailed that.
But they aren't attracted via a hydrogen bond, are they? Like, they don't have hydrogen in them. How could they? I'm going to say A, 0.
All right. Another drum roll. Or you can, this one is 3.
3?
Yeah.
Oh, how?
Yeah, well, that's what we're going to get into.
Okay.
But I believe there is one more question that relates.
Oh, let's go. I know this one.
Yeah. Well, hopefully. So, what is a covalent bond? Is it A, a type of dipole-dipole interaction? B, a chemical bond formed by oppositely charged atoms through a full transfer of electrons? C, when 2 atoms love each other? Okay. Or D, a sharing of valence electrons between atoms?
It is D, the sharing of the valence electrons between atoms.
And you are absolutely correct. It is a sharing
Let's
of valence
fucking
electrons.
go.
So, yeah, then there's a point of clarification. So, going back to, so you nailed C with G and A with T when it comes to, typical, healthy base pairing is what we call it. Okay. But what holds the A's and T's and the C's and G's together are the hydrogen bonds. As in, if you look in between, let's say starting at the top, okay, A and T, the dashed line, or actually maybe I should go backwards. Go back to the original. Gonna have to go back a few. Oh, I was there. How long ago? Yes. The nitrogenous bases.
Yes. Okay.
So we were right. Yeah, that's where. So this is how the base pairing occurs. Okay. So the main structures. Okay, but the hydrogen bond is right there. That's what holds. That's what holds the C's and G's and the A's and T's together. And you have your double-stranded DNA that we'll get back to that figure. But that's how the base pairing occurs. A with T, C with G, and A and T has 2 hydrogen bonds, and C and G has 3 hydrogen bonds, those 3 dashed lines. And as you can maybe make out, hopefully, that
It's
there
stronger.
yeah, exactly. The interaction, the base pairing between C and G is absolutely stronger, and that is actually pretty key and important. And hopefully you can see that there is hydrogen sort of like sandwiched.
Yeah.
Okay. And that's what makes it, the hydrogen bond. Where you have a hydrogen, let's say, looking at the C and G one, you have at the top, you have hydrogen, and on the left is an oxygen, and on the right is a nitrogen. Those are 2 electronegative atoms, and that results in, a hydrogen bond. But we're going to be covering it more and more. We're going to be seeing a lot of hydrogen bonding as we progress throughout some biochemistry. So that's how the base pairing occurs. So now getting back to the fancier...
Fancy time.
Yes. Right after... Clear the bond.
Was there another question? All right.
No. The questions are over for now. Yeah.
Okay.
So I just wanted to show this again of you have your nitrogenous bases, okay, your 3 components of the nucleotides that you nailed. You have your pentose sugar, okay, and we call it deoxyribose in DNA. That is the pentose sugar. You have your phosphate, okay, and then you have your nitrogenous base, and those nitrogenous bases are the A's, T's, C's, and G's. And that's what we mean, that sequence of A's, T's, C's, and G's. That's what we mean when we talk about sequencing your DNA, of that sequence of A's, T's, C's, and G's. And you have the base pairing. It's the hydrogen bonds that allows them to base pair, or to effectively stick together to create those 2 strands that, double helix up. Make sense so far?
Yep.
All
Makes sense.
And so now getting into some newer stuff, just as, a little bit of information about DNA sequencing, okay? Where, on the right there, the cost of DNA sequencing has gone down over time, but obviously, we're at 2024 now, so it's probably even a little bit less, okay? And, that's what we mean. So what you're on the left there, as they say, sequencing the human genome back then was a daunting task, cost, tens of millions, hundreds of millions of dollars. Now we do it many times cheaper. And in the human genome, okay, it's basically about 3 billion base pairs, okay? And when we say base pairs, we mean those A's, T's, C's, and G's.
Nitrogen bases. Pairs of nitrogen bases.
Exactly. And so they're showing you a little bit of a DNA sequence, okay? Yeah, did you have a question?
No, I was going to say, so then there'd essentially be at least 6 billion G's, T's, A's, or B's.
Yeah, there's exactly. There's
paired.
Yeah, nailed it. Exactly.
Okay. Interesting. That's a lot.
And then, as we'll see, the DNA gets transcribed into RNA, gets translated into protein. And they're just showing here a little bit of a snippet of the DNA sequence that encodes for...
Are they just fucking DNA docs, buddy?
Yeah, exactly.
Fuck, oh shit. There's a lawsuit.
So, now getting into some of the newer stuff. So now we're getting into protein structure and composition, okay? We've covered the fundamentals of DNA. We're going to be covering it again. We did not do a super deep dive, but, in a little bit, we'll get there, of a super deep dive on DNA, because there's more to DNA than what we just saw. But to put it in context, okay, I've mentioned the central dogma many times before, okay? And just to reiterate or remind, the central dogma, I don't like the terminology, but it says DNA gets transcribed into RNA, and then RNA gets translated into protein. Okay, that's sort of like your flow of information in the biology world. And so as I show there in the bottom, DNA encodes for protein, okay? And the analogy that I like to use, as I put there in the slide, is if all of biology is a movie, DNA is the script and proteins are the actors, as in they do the stuff. That's what you see on the screen. That's what that's what does the actions, or pretty much the vast majority of actions in biology is the protein stuff. It's not all protein, but proteins, I would say, are like the key players in, as they say there, in virtually all biological processes. So understanding proteins is super important, and so we're going to be introducing that today. So getting into that, before diving into amino acids and proteins, we need to, I guess, introduce or get familiar with how we represent things, because we're going to be looking at molecules, we're going to be looking at amino acids, and so I want you to be able to at least get a sense of what we're looking at, or start to understand, because there's multiple ways that we depict molecules. Where at the top there, okay, you have a water molecule, okay, H2O, and so we have our typical, at least in the biochemistry world, have our typical color scheme of oxygen is typically shown in red, carbon in gray, nitrogen in blue, and sulfur in yellow, and hydrogen in white or like light gray. Apologies to anyone who's colorblind out there. But
yeah, that would suck. I wonder how that works. I guess you just look at a different thing. There's feedback, quite a bit of feedback coming through. I'm just gonna mute. Okay, I'm gonna just mute.
Yeah, take your time.
It might be coming from.
If it's me,
no, it's all good. I muted it. It was coming through the PowerPoint.
Gotcha.
Oh, it's good though.
But yeah, no, it'll take some time getting used to, but absolutely everyone will get used to it. It took my brain some time to, get used to just looking at molecules and like, what am I looking at? Well, what are these atoms, etc., etc. But your brain absolutely will adjust. It just takes some time. But yeah, just that's our typical color scheme, at least in the biochemistry world. Whenever you see like a red sphere or a red ball, that's oxygen. Where we see a blue ball, that's nitrogen, and yellow is sulfur and carbon and gray. So here is where I can say, with that in mind, we're now in chapter 2 of the textbook. Okay.
Shit.
Protein composition and structure. Okay.
Oh god damn, it's intense.
And we're gonna be starting off, zoomed out. Okay, giving you just a broad overview of proteins, but then we're going to, start zooming in, and I'm gonna try and go, methodically, step by step, and like absolutely with time, you'll get used to like seeing the molecule, seeing proteins, and I'm always gonna try to be putting in the context of, again, we're talking about, when we're talking about proteins, it's like, well, DNA goes to RNA, goes to protein. So when this protein was made, well, before that, started as RNA, and then before that started as DNA, started as like a genetic code. But we're going to, see that in much more detail later on. But I just want to put that context. So, getting into protein composition and structure. Composition and structure. So, key point here, as I highlight, is structure determines function. Okay, that's a common theme in biochemistry. That's a common theme in biology overall. That's a common theme for life. Of like the structure of a thing, the structure of a protein, like determines its function. And protein structure is critically important, not only to understand them, but it also has medical applications, because like virtually all diseases and conditions, relate to protein at some level. But protein structure can be simple and small, like shown here on the left. Pretty much just like a helix, just a little coil, winding around. Or it can be these large, elaborate, what we call, macromolecular complexes. So there's like a bunch of stuff going on that, understandably, looks overwhelming. Like even to me, looks overwhelming. We're going to see how these are made. But again, at some point, okay, the figures here, they started off as, something in the genetic code, as in some sequence of DNA, okay, coded for this sequence of amino acids, okay? Now we're going to be looking at amino acids shortly. But again, we're starting off zoomed out. So, proteins can form interactions with one another along with other biomolecules. What does that mean? Well, pretty much, so proteins can be entities in and of themselves, okay, where you can have protein number 1, okay, you can have protein number 2, but then very often, you know, protein number 1 can like interact with protein number 2, or they can like actually come together and, you know, sort of cooperate, so to speak, and, you know, carry out a bunch of functions. But we're going to get to that with time. I'm just introducing the concept now. We're shown here in the figure is basically muscle, well, protein that you see in muscle, and we're going to look at some point of like, well, how do these proteins, the way they're assembled, how do they generate force like in muscle? Now we're going to see that, you know, proteins can undergo dynamic movements, okay? They're not just like these Lego blocks that are just like, you know, rigid and do nothing. Sometimes they are rigid because we want them to be rigid, but very often, more often than not, proteins are highly dynamic. They undergo like movements and stuff like that we're going to see. But again, I just want to introduce the concept of like, yeah, they can be simple, but they can build up to something super complex like a muscle fiber. And they're zooming in on a muscle fiber there in the figure. But again, at some point, we're going to be looking at that in detail as well. And then, yeah, another sort of zoomed out look is, again, just as I mentioned, they can be dynamic. And proteins, not only can proteins interact with each other, they can interact with other molecules, okay? Where shown here in yellow, so in yellow, this is one way of depicting a protein, okay? The protein here is called lactoferrin, okay? And it binds iron, okay? This is actually one of the ways that we kind of like store iron in our bodies, sort of bound up into protein. But what they're showing here is that when this little iron molecule, or iron atom, okay, binds lactoferrin, there is what we call a conformational change, as in, you know, sort of like the lactoferrin sort of like engulfs it, or pretty much there's movements that happen when the little iron thing, you know, binds to the lactoferrin. And that's a common theme that we'll see. But as pretty much, it's not a binary, you have all the entire spectrum of like something that's highly dynamic and flexible, but then other proteins that are rigid, okay? Like, let's say, for example, your hair and fingernails, okay? Those are proteins, keratin. And so your fingernails, quite rigid, okay? But other protein in your body, they'll be, you know, quite flexible. But all that to say is just to introduce the themes. So like you can go from rigid to flexible. And there are conformational changes that can occur. So that's the zoomed out, okay, version of proteins, but we're not quite there yet, okay? I just want to introduce these pictures and these figures and these themes, but to fully understand proteins, we have to understand amino acids, and that's what we're going to start diving into right now. So,
Goddamn.
This is an amino acid, okay? Where the building blocks of proteins are amino acids, okay? So, and these are these molecules here, okay? So you have this R group, okay? This changes for the 20 amino acids. So basically, we're going to be looking at 20 building blocks, okay? And one part stays the same, okay? As in, you'll pretty much always have this COO here, what we call the carboxyl group. And you'll always have this amino group there. You know, that's why we actually call them amino acids, because you have this amino group here. If it's not clear yet, it will be, you know, again, we're going to be taking this systematically as we go. But this is your overall structure of an amino acid, whereas we're going to see everything will stay the same, okay? You're always going to have this COO group here. You're always going to have this amino group here. And you're always going to have what we call this C alpha in the middle, what we call the alpha carbon, okay? But this R group will change, and that's what makes the difference in the amino acid, as you will see. We're pretty much going to go like, almost like one by one, of like amino acid number 1, amino acid number 2, and we're going to see what they look like. And you will pretty much, they'll be imprinted on your brain. You'll get familiar with amino acids. But, shown here in this figure, there's two versions of amino acids. As they say there in the figure, they say it L isomer and D isomer. And you might be asking, well, what's the difference? Or like, who cares? Yeah. Well, just as let's say you have your left hand and you have your right hand, okay? But if I ask you, and just as you have your left shoe and your right shoe, but do you enjoy putting your right shoe on your left foot and your left shoe on your right foot?
I don't. It doesn't feel good. It's just wrong.
No. And just generally speaking, unless you have the most simplest of gloves, your left-handed glove goes on your left hand, and your right-handed glove goes on your right hand. And the same thing holds true at a molecular level or an atomic level of actually the three-dimensional orientation, okay, actually matters. Where it's a little bit, these L isomer and D isomers, at some level look identical, but they're actually not. You can think of it as like one is left-handed and one is right-handed, okay? Or like if, this is getting into organic chemistry, but hopefully you can see that if I draw a line down the middle, okay, between the two of them. And I
mirrored essentially.
Perfect, exactly. You nailed it.
Yeah.
They are mirror images of each other, just as your left hand is a mirror image of your right hand. And your left foot is a mirror image of your right foot. And that matters, okay? And we're going to see that at some point of just the three-dimensional orientation of, even though these have pretty much like the exact same atoms, the fact that they're oriented just slightly differently in space actually sometimes has very dire consequences.
Uh-oh.
But the key point here is that pretty much we can ignore the D isomer. We only find the L isomer that are found in proteins and pretty much like in nature. So we're pretty much only going to be looking at the L isomer. But I just want to highlight that, three-dimensional orientation matters. Just introducing that concept. But you can pretty much ignore that for now. We're just going to be looking at, the left-handed version of amino acids.
Oh, yeah. So, as a left-handed person, I appreciate that. We don't get enough respect or understanding.
Absolutely not. So, diving into, systematically, amino acids mostly one by one, okay? Where this is just one amino acid. So, glycine, okay, is the simplest amino acid. Where the R group there, okay, R is not an actual atom on the periodic table, okay? That's like a placeholder, And so in this case, to actually be an amino acid, okay, the R group is replaced with a hydrogen atom. That's what makes it glycine. So we call it glycine. Everything else is the same. You still have NH3, that's our amino group. We still have our carbon there in the middle, the alpha carbon. And then we have COO minus, and that's our carboxylic acid group. And that's why we essentially call them carboxylic acid. And so if you have hydrogen there in place of the R group, then by definition, that is glycine. And your two-dimensional version of glycine is shown there at the bottom. Your fancy, ball and stick with the color-coordinated version. That's shown there at the top. Where again, the color scheme, oxygen is typically shown in red, carbon in gray, and nitrogen in blue. So that's your glycine amino acid. It's the simplest, okay? And we're going to get more and more complicated as we go. Just starting with simple and moving up in complexity. And then the next, amino acid is alanine, okay? And pretty much everything else is the same, except now the R group is different. Instead of a hydrogen atom, you have a CH3. You have carbon with 3 hydrogens, okay? And you have your two-dimensional version of alanine there at the bottom. And you have your fancy ball and stick, still the same color scheme. And we call this a methyl group. That's just fancy organic chemistry terminology, okay? But that's why we call it alanine. Like in that position, when you have CH3, that's an alanine. So hopefully you're seeing the theme of amino acids.
Yeah.
But still going, building up a little bit more complexity, okay? Where now we have going 3 amino acids at a time, okay? Where you have proline, valine, and leucine. So now we're getting into the fancier R groups, a little bit more complexity. Still, the fundamentals are the same. You have your NH3 group there. you have your alpha carbon there in the middle, and in purple that I've circled, or in an oval, that's where I've highlighted the R group. That's our R group there. And we're going to see that these have, certain chemical properties, where so far, all of these are just, a bunch of carbon and hydrogen.
So,
in the R group. Go ahead.
In proline, the hydrogen in the proline isn't in the R group.
Exactly. That's like just jutting.
It's just
out.
around it. Yeah.
Exactly.
Okay.
Exactly. Just as if you look at the other ones, they also have a hydrogen. It's just like, it's sticking out a little bit differently.
Oh yeah, I could have looked at the picture better.
No, it's okay. I understand it might be a little bit too small. But yeah, proline stands out because like its R group is like, backtracking like on itself. Okay, as opposed to just like, going straight out there. But we're going to be taking a closer look, at proline and, how these, interact with each other. Where, as of right now, we're looking at the building blocks, okay, of proteins. The amino acids assembled together, as in I think of amino acids like the Lego blocks of protein, and we start assembling them together, okay, in various combinations, pretty much infinite combinations, that we build up into our complex proteins. But yeah, as I mentioned there, the note, so far all the R groups, okay, and we refer to these now as side chains moving forward, where just like the side chain of the amino acid, everything else stays the same. But so far we've just seen made up of carbon and hydrogen, but they're going to get a little bit more complex as you go. They're not going to get super complex, okay, but just slightly more. So moving along in other amino acids, so yeah, now we get into isoleucine, methionine, tryptophan, and phenylalanine. So again, still overall the same scaffolding. You still have your amino group, you still have your alpha carbon there in the middle, you still have your COO minus, and again, in the purple oval or circle, that's where I've circled the R group, the side chain. Okay.
Ooh. Yeah. I like phenylalanine. The H fucking little hydrogen circle's fun.
Yeah.
Oh, the other one's got multiple.
That's exactly. Cool. Multiple ring structures, and these also have like special properties, so to speak. But their side chain is getting a little bit more complex. Where now you have like this ring structure in the side chain of phenylalanine, and you have these double ring structure in tryptophan. But that's its side chain, and we're going to see that since everything else is held the same, like it's the side chain of the amino acid that that's what gives it its special properties that we're going to get to.
Never show a Christian or a young Earth creationist, has to be more specific, the fucking structure of the methionine at the very bottom that looks like a cross.
Oh, true. Oh, my goodness. Good.
No, I know about the protein laminin. Is it a protein?
Yeah, exactly. Laminin.
I only know about that because it looked like a cross in one picture, and it has been used by many Christians as like a proof that God is even in our protein because laminin structure looks like a cross.
When we depict it, it's sort of cartoony, but we're going to see at a molecular level, like, you're not going to be seeing crucifixes or crosses when we zoom in like that.
Yeah.
Just as like, that. if we depict it like this, but if we start incorporating like the hydrogen atoms and all that stuff, okay, we're simplifying it here, where it looks like a cross. But
well, even looking at the picture at the top, like it doesn't look like a cross at all.
Yeah, exactly. Exactly. The one at the top is more like the more accurate molecular atomic representation of we're incorporating everything.
Well, we didn't know that.
No, I wouldn't expect you. Totally not your fault.
But yeah.
And so, these are the side chains. So again, we're building up. So far, I think we've covered about 10 or 11 amino acids. Okay, so we're about halfway through. But so now serine, threonine, and tyrosine. So again, pretty much everything else stays the same, but now the side chains are different. Just want to introduce them. And nothing overall super fancy here. Tyrosine also has another ring structure in its side chain. Yeah. Very similar to phenylalanine. Okay. And I don't expect you to have these memorized. I mean, I'm sure at some point you will, because we're going to be seeing them over and over. Yeah, but at some point, I had to memorize all of these. I had to on request, pretty much eyes closed. If they tell you an amino acid, I gotta like be able to like draw it like by hand.
Yeah.
Everything. But even today, even though I memorized it back then, like I couldn't. I definitely would not get 100% on all of these. I would get like the major points for like most of them.
Yeah.
But like I would miss like some small details on like the more complex amino acids, like the phenylalanines, like the tryptophans, and all that stuff. But like,
Tyrosine looks fun.
Tyrosine is, and actually we're going to see that tyrosine is sort of a little bit of an MVP when it comes to amino acids.
Let's go.
And then we have asparagine, glutamine, and cysteine. Again, nothing overly fancy here. Everything pretty much, again, just showing you the different side chains and or R groups, as we say. Where now cysteine, cysteine has a sulfur, and that stands out. Whereas something a little bit different that I don't think we've seen before, but everything else is more or less similar to what we've seen
yellow.
before. Exactly. Where that's our color scheme of sulfur, typically depicted as yellow. But glutamine and asparagine, and as we're going to see, these aren't the way that they're named. It's not fully coincidental. As in, there's a reason why, let's say, glutamine, it ends in amine or ine, and there's a reason why asparagine also ends in ine. Although cysteine also ends in ine, but that's all. That's actually mostly coincidental. But all that to say is, let's say, asparagine and glutamine sort of have a little bit of a relationship to each other, where hopefully you can maybe see or make it out, like in the side chains, what's in the purple circle, like not super different. There's actually some similarities. Or it's not like a world of difference.
Yeah, they're gonna hang out.
Yeah, exactly. And they very often do hang out. And
it's like they have a Pokemon Altaria on the top of them.
And then sort of, I think covering the last 5 or so, we have these 3, and I think 2 left. Where, lysine, asparagine, and histidine. Where again, just different side chains. Histidine stands out a little bit as it has a little bit of a ring structure in its side chain. And also histidine is also a little bit of a MVP when it comes to amino acids.
Let's go.
And then lastly, the last 2, aspartate and glutamate. And these are also, it's not a coincidence that these are coupled up together. Where again, if you look at the side chain or R group of what's in the purple circle, it's not super different of what's in there.
Is it a coincidence that aspartate sounds like aspartame?
No.
Is that a coincidence?
No, it's not a coincidence at all. Where aspartame is essentially a derivative, of aspartate. Where actually, I should probably, I'm going to add that as a note to hopefully incorporate that next time to show you, I'll show you aspartate again and show you aspartame, and you're going to see that aspartame looks quite similar to aspartate, or aspartame is derived.
And glutamate, like glutamine?
Yeah, exactly. There's a relationship between glutamate and glutamine, just as there's a relationship between aspartate and asparagine. It's all
I was gonna make the asparagus asparagine thing before, or whatever, the one before, and is there actually a connection between that? Do you know if
I don't think so. There might be some sort of like loop, but I think it's mostly coincidental. But no, awesome question as always. So that wraps up, the amino acids, like the structures. I don't expect you to like memorize them, but we're going to be seeing them over and over. So at some point, they will be tattooed on your brain. I just want to introduce them.
Let's go.
But now, I just want to introduce the concept that we saw 20 of them, but they can be grouped together, or they can be categorized, in a certain way. And this is one of the common ways, very common ways, that we group them. I don't expect you to understand this grouping now, because we're actually going to get to that in the next few slides. But I'm just introducing it now. Like, what does it mean when we say, aspartic acids? Aspartic acid is another way of saying aspartate. Glutamic acid is another way of saying glutamate. And the rest is the same. What does it mean when we say acidic or negative charge? What does it mean when we say basic, positive charge? uncharged polar. Yeah, go ahead.
Is that the pH levels, or is that different?
Yeah, no, that we're totally going to be talking about pH probably next time, or maybe we'll get a little bit
Let's
to it this time.
go.
But like you're absolutely right. It has everything to do with pH. And yeah, uncharged polar, and this big grouping here in green, the side chains non-polar. So we're going to get to like, well, what does that mean? Non-polar, uncharged polar, basic, positive charge, acidic, negative charged. Just 1 second to get my question from my own chat. Which are the ones that we still have trouble creating through? Good question. 1 second.
Okay.
All right, moving on up. So I just want to make sure. Yeah, that's the next slide. The amino acids. Okay, so going a little bit more systematically. So yeah, physiological pH. So like you nailed it. In terms of it absolutely has something to do with pH. So what does it mean when we say, it's acidic? Okay, well, the side chains of aspartate and glutamate are very similar, both structurally and chemically, where they both carry a negative charge at the end of their side chains at physiological pH. Okay, more on pH later, or probably more on pH next lecture. But yeah, the only difference is glutamate has an extra CH2, red circle, in its side chain. And hopefully you can see that, like pretty much glutamate and aspartate, virtually identical, except glutamate just has that one extra CH2, that red circle there. That's all. That's the only difference between aspartate and glutamate. But the key thing here is like, why do we group them like this? Acidic negative charge? Well, if you see at the end what I put in the purple circle or oval, there's a negative charge there, okay? And so that's why we group them like that. These are the amino acids that they have that negative charge in their side chain, okay? Everything else stays the same about the amino acid, the NH3, the COO minus, blah, blah, blah. But like,
is it important that the fucking O in the left are on the aspartate is on the left and on the glutamate it's on the right?
It is sometimes consequential, absolutely. Where going back to the theme of like the three-dimensional orientation of atoms, like matters, and just as like left-hand, right-handed version matters, like it is sometimes consequential. And even though glutamate and aspartate are very similar, the only difference is like what I put there in the red circle, just that one extra addition of CH2 like makes a difference. There's a reason why glutamate even exists in the first place, where sometimes it doesn't make a difference, but sometimes it does and is super impactful. And then revisiting more of the groupings of the amino acids. So we covered the negative charges and the acidic. Now we're covering the basic or the positive charges, okay? And just again at physiological pH, but we're going to get to what that means, in the next lecture, next week. But all that say, we're grouping arginine, lysine, and histidine in this way because in their side chain, okay, they have a positive charge. We saw that aspartate and glutamate, they have a negative charge, okay? Histidine, arginine, and lysine, they have a positive charge, okay, at their side chain. Now, as I showed there, or as I mentioned there in the note, the positive charge on histidine is not shown, but it is generally there, okay? They're just not showing it for whatever reason. But as I say, they don't always have a positive charge. Rather, under physiological conditions, i.e. the conditions of the body, they have a positive charge. We're going to see, if it's not that, I don't expect that to be clear now, but it will be clear probably next week. We're going to be revisiting this over and over. But all that to say, when we're talking about, let's say, the amino acids that you have in your body, okay, that are, let's say, maybe circulating around, let's say, in your bloodstream, they will have a positive charge. but we're going to see if, let's say, we take them and let's say we put them like in a beaker under different conditions, that
Mhm.
positive charge might be
Would
gone.
go away.
Exactly. But again, we're going to get to pH next week.
Could it go negative? Could it get acidic? Maybe under certain situations.
Yeah, but that would be like be really extreme basically, you know, hypothetically yes, but we effectively in reality don't really see that, but hypothetically it could happen. Good question. Yeah, definitely have to think about that one. So then the other groupings, okay, the uncharged polar. Okay,
They're just chilling.
So yeah, these amino acids do not have full charges in their side chain. So we saw aspartate, glutamate, they have a negative charge in their side chain. And we just saw on the last slide, lysine, arginine, histidine, they have a positive charge in their side chain under physiological conditions. Just 1 second. So they have polar side chains. That is, they have partial charges on their side chains. Okay. And so cysteine is missing from the table because it is considered mildly polar, where again, the groupings and all that stuff, depending on the textbook, okay? The table that I'm using is from a different textbook.
Yeah.
But all the molecular structures, that's from our strictly biochemistry textbook that we're primarily
Mildly
using.
polar, would that fall on the positive or the base or the acidic or either?
Good question. And that's what we're going to be clarifying in a moment.
Oh, shit.
So yeah, you're asking the pointed questions.
And that cysteine night, I can't really see. Cysteine?
Yeah, cysteine. Yeah, cysteine.
Cysteine, like the Sistine Chapel.
Yeah, that's actually a good way to remember it. We're gonna be covering polarity. We've covered it a little bit, but now we're gonna do a little bit of a deeper dive, or just to remind you of what do we mean when we say, you know, polar. What does it mean to be polar? You technically do already know, but it's not obvious to you yet. But it will be in a couple of slides. Oh, yep. So, and then lastly, okay, the last grouping of amino acids. This is where it's, I would say, the simplest. These amino acids have no charge at all. Okay, where basically, if you look at the side chains, the R groups, okay, in the figures there, it's mostly just a bunch of carbon and hydrogen. Okay, and we call them non-polar or hydrophobic. If you want to maybe take a guess as what do we mean when we say hydrophobic? If you want to
They're afraid of water or repel water or like go away.
Exactly. Where they repel water or they are afraid of water. Basically, they don't want to be under water conditions. Okay, and that actually matters, as we'll see. But yeah, pretty much these are our non-polar / hydrophobic amino acids. As in, these amino acids pretty much don't want to be exposed to water. And that has, biological consequences.
Oh shit, we can toss some water on those bad bitches? Oh my goodness.
We just might. Wrapping up this lecture, we're gonna end with polarity, and as a reminder, probably gonna start with polarity next time, just to review it. So what does polar mean? What do we mean as biochemists when we say, you know, these amino acids are polar or uncharged polar that we saw on the table? Well, we've seen this water molecule, right, before, okay? And we've seen that, you know, you have this partial negative charge, okay? And you have this partial positive charge on the hydrogen, right? You remember dipoles and all that stuff?
Yeah.
Where oxygen is more electronegative. It's greedy with electrons, okay? And therefore, it's trying to pull electrons away from hydrogen. And therefore, it gains this partial negative charge, okay? And then you get this partial positive charge on the hydrogens. So, as I say there, since the water molecule has partial positive and negative charges, it is a polar molecule. But, okay, doesn't end there. The 3D shape of a molecule matters, okay? As we've alluded to before when it comes to left-handed amino acids and right-handed amino acids. And so, why does it matter here in this context? Well, shown there is carbon dioxide, CO2. carbon in the middle, and 2 oxygen atoms on the side. So you still have, a partial negative charges, on your oxygen atoms, on the left and the right-hand side. And you have a partial positive charge on the carbon in the middle because, again, oxygen is greedy with electrons. So
greedy. Oh, fuck, I hate it.
Exactly.
Carbon the capitalist.
Yeah, and so carbon can't hold on to its electrons because it's being pulled from both sides on the oxygen. And so, yeah, you end up with a partial positive charge on the carbon and partial negative charges, on the oxygens, But despite having these partial positive and negative charges, carbon dioxide is nonpolar. So what's the difference? Well, that's what I'm going to clarify now, hopefully. We're wrapping up, or I think next slide we'll wrap up. What does polar or nonpolar mean? Well, it's a tug of war for electrons. So as we saw with a water molecule,
Yeah, I was gonna ask why fucking carbon's getting like hit from both ends, but fucking oxygen's taking it from like it's like coming from the bottom and not on the side like it.
Yeah, so you're asking a really fantastic question.
Well, that's what it looks like.
Yeah, you're pointing out like the key difference, and actually now that you I'll probably add a slide in for next time to clarify even further, like why is it that oxygen's like that, and why is it the carbon's like that. But let's say for now, the fact that carbon dioxide is linear, and the fact that that water is sort of like a triangle, then the tug of war for electrons plays out differently, where in water, since oxygen is more electronegative than hydrogen, oxygen wins the tug of war for electrons, and you still have the same tug of war in carbon dioxide. Since there's oxygen on each end, nobody wins the tug of war, as in there's no net dipoles, or carbon dioxide is nonpolar. What do I mean when I say no net dipole? That's what I'm going to clarify in the next slide.
Is it because it's positive on the ends and negative on the ends?
Close, where the tug of war, you can think of it as, since the tug of war is opposite, as in opposite directions, nobody wins. As opposed to since it's on an angle, in the water molecule, oxygen ends up winning. Where I'm sort of showing that they're canceling out, as in since in carbon dioxide, the oxygen on the left, is like in tug of war with the oxygen on the right. And since they're perfectly on a straight line opposite each other, they cancel each other out, But there's no canceling that occurs in the water molecule, where the arrows are pretty much all pointing towards the oxygen, because oxygen's in the middle, and the hydrogens are too weak, it cannot compete. And so like the forces are going in the same direction towards the oxygen in water, as opposed to in carbon dioxide, since it's straight, and linear, the forces are going in equal and opposite directions, and so they cancel each other out. Like literally like a tug of war, if you have two people of equal strength on opposite ends of the rope, and they're pulling with the exact same strength, nobody wins, right? But if I take the same scenario of a tug of war of two people of equal strength in the middle, and then I take a third person and they start pulling in the center, that third person's going to win, because nobody's canceling that person out if they just pull from the center of that tug of war. If that analogy makes sense. So just as a magnet has a north and south pole, molecules have polarity. So to be a polar molecule, you need partial positive and negative charges and a net dipole moment, So in the water molecule, you do have a net dipole moment. You have partial positive and negative charges, You have a net... Yeah, go ahead.
Oh, sorry. Maybe you're just about to say this. A net dipole moment is what, when they're pushing together towards?
Yeah, exactly. Where pretty much you have no canceling out. As in sort of as I've depicted there in carbon dioxide, since one arrow is going left and one arrow is going right, they just like cancel each other out, and so you have no net pretty much nobody's winning the tug of war for electrons. That's ultimately what we mean when we say no net dipole. As in, like, the water molecule, it is, water molecule is winning the tug of war for electrons. And so you do have a net dipole moment, okay? And so, yeah, in carbon dioxide, the dipoles cancel each other out, so there's no net dipole. And so that's polarity in a nutshell, but we're going to be revisiting this, over and over. We're going to be revisiting amino acids over and over and over. So yeah, if it's not clear now, like, I have no doubt that it will be clear. I just want to, introduce these concepts now. And again, we're building up, okay, to, proteins. But I think this is, like, a good place to stop, and I also want to, like, respect your time and not overwhelm with too much. I think we covered actually quite a bit of ground today.
I think I get quite a bit of it. I think I get the shape difference in those types of things. I think I get a bit of it, but I don't fully understand the net dipole movement or moment. But I'm sure we'll learn more, and it'll be more clear as we go. But I feel like I get the rest, and the rest makes some sense.
Good. And well, the DNA question at the beginning, like, you got 3 out of the 4. And so, like, and there's been a 2-week gap, right? So, yeah, you are absolutely learning. And yeah, you asked a great question. And now that you asked it, like, I'm gonna add it. I should add in the slide for next time. But, like, yeah, you asked a great question of, like, why is it that oxygen, it's shaped the way it is? You're basically, why is water not straight and linear like carbon dioxide, where you have 3 atoms in the water molecule, you have 3 atoms in the carbon dioxide, CO2. And so, yeah, it does beg the question, which is the great question of why is it that oxygen, is like that, sort of like angled out, and carbon dioxide is just, like, straight and linear, like that.
Can you get an... Oh, sorry, I don't mean to cut you off.
That's okay.
But can you get an angled one like hydrogen? I know that it wouldn't work with hydrogen and oxygen because of the amount of bonds, but could you get a double bond and still be angled like that?
Yes. Well, not angled well,
like, not necessarily be, like, look like a fucking bop it, like, fucking be angled a bit. Doubled.
Yes. Hypothetically, yeah. You can have well, I'm not gonna say just about any, but the short answer is, like, yeah, you can get double bonded and angled like that. It's less likely, but it can happen. But totally, the fact that you have that double bond in carbon dioxide, that is one of the major driving forces as to why it's linear, and that you don't have the double bond in the water molecule. There's something else going on in the water molecule that is not depicted there that results in it being angled like that, and that's what I'm going to try to fill in next time. But, like, yeah, why is it that pretty much what is ultimately happening is, like, yeah, the hydrogen atoms are being pushed down, they're being scrunched down.
And that double makes it harder to push because
Exactly.
it's a thicker, like, gnarlier fucking thing to push.
Exactly. But
it's like one of those bendy bars.
Yeah, exactly right. And there's a reason why the hydrogen atoms in the water molecule are being scrunched down. It's just not obvious here. It's not shown at all here. But, like, yeah, I will show it next time. That should clarify it. But yeah, that is the good question. That is, well, the question. But yeah, given the fact that, like, the hydrogens are being scrunched down, that's resulting in, that angular. But yeah, there's a reason why they're being scrunched down. But yeah, if the net to dipole thing isn't clear now, don't expect it to be. But even if you don't ever fully get it, but I know that you will, it's not like the most critical thing to understand what's going on in biochemistry. Other than, like, I think the critical thing is knowing that dipoles exist, and, like, it's driven by electronegativity. I think those are sort of the critical components. But I have no doubt, once I provide more detail into what's going on here, you will get the overall net dipole aspect. But I would say think of it like a tug of war, and that ultimately, if some atom is winning the tug of war of electrons, then there's a net dipole. If there's no atom winning the tug of war, then there is no net dipole. But again, I have no doubt that it'll be clear in time. So yeah, that's all I pretty much had prepared or in mind for today. I think we covered, again, quite a bit of ground.
Oh yeah.
Did you have any questions, feedback, criticisms? You want
No,
to drop the course?
no, I certainly do not want to drop the course. I'm stoked and I'm very excited, and it's always the most exciting time. And every time I learn shit like fucking just like a little bit more, like I feel like my brain opens a bit more, and it's like, oh, cool. That is super interesting and fucking super fucking stoked. I love the fucking questions. More and questions are always happy. I like questions to like know if I actually understand things. And like that's one of the best parts about doing something like this is like having like kind of like tests or like questions or being able to ask questions. Um, because yeah, like you think you understand or know something until like you're asked a question about it, and they're like, "Oh yeah, no, I have no clue." Or you don't think you understand something, and then you're asked a question about it, you're like, "Oh, this," and then it's like, "Oh no, I definitely do." So it's really good check of knowledge and to see if it actually sunk in. So I like that.
Good. I'm super glad. I'm gonna try and incorporate more questions as we
That's
go.
good stuff.
And like, yeah, I would say so far you're pretty much like nailing virtually all of the questions. Like, you definitely remembered the nucleotides. Okay, what makes up the three components of a nucleotide? And hopefully now it's a little bit more clear of like what do we mean when we call like base pairing, the C's and G's and the A's and T's, and the hydrogen bonding. So absolutely, you are learning.
Yeah,
So we'll pick
appreciate
up
it.
here next
Doing
time.
great.
Sound good?
Yes, thank you. Well, have a good one. I'll see you next Wednesday.
See you next Wednesday. Take care. Be
All right.
well. Always a pleasure.
Yes, you as well. Much love. Bye.
One moment. Well, first of all, Jacanadar and Bob Marley and anybody else that I forgot. One second. Orange You, thank you for passing by. And yeah, definitely the dipole stuff. But again, he will get it. I have no doubt. No doubt about that. Just I have no doubt that anybody will get it. If they attend regularly, since we're going to be revisiting these things over and over and over again. Just one second. But yeah, the dipole stuff and all that is not like, the most hardcore of biochemistry stuff. But it is still, there's still some importance to it. All right. But let's see. I apologize if I missed some of your comments. Yeah, oh yeah, answer that. Histidine, kind of the oddballs, and yeah, the imidazole group. You are remembering that correctly. That is histidine, and yeah, that imidazole group. That has consequences definitely when it comes to, research and all that stuff. And histidine and purifying proteins, etc., etc. Essentially we exploit histidine amino acids when it comes to protein purification, but we'll get to that in due time. And you are using, I guess you have your own biochemistry textbook, I imagine. I'm curious as to which one you would have. Okay. I need a little bit of hydration. One, because I've been talking a lot. And two, because we're in a little bit of a heat wave here. Selenocysteine and pyrrolysine, that's probably not for a decent amount of time. Yeah, that's probably like 10, 12 chapters from now. Those are pretty niche modified amino acids. But we will eventually get to them. We're in chapter 2. We're officially in chapter 2 of the textbook. And I think there's like 30 chapters in total in the textbook. Or 36? Let me double check. Oh, whoopsies. Well, Google Scholar definitely has some good stuff on it. And yeah, if you're reading on it constantly, all the more power to you. I definitely enjoy reading textbooks myself. There are... yeah, 36 chapters. So, you know, we're like 6% done the textbook. Although I think some of the chapters, you know, right now we're in like the really dense stuff. And also, you know, yeah, in addition to like covering the textbook, I'm also like trying to cover, you know, some other theory as well. As in, like, obviously I'm covering some chemistry aspects as well, like the dipole stuff and polarity. It's not covered in the textbook because it's assumed that you know that from previous courses. But as I continue to lay down the groundwork, you know, to understand biochemistry, then I have no doubt that, you know, the pace will pick up in terms of like covering more textbook material. Right now it's like, you know, 50% textbook and like 50%, you know, covering the fundamentals to understand what's going on in the textbook. But as that foundation gets more and more built, then I have no doubt, or almost certainly, it's going to be like pretty much 100% focus on like what's going on in the textbook. Oh, if you keep up with phylogeny, then yeah, also all the more power to you, because it's definitely not my area of at least high interest. But let's see what I'm going to do. As a little bit of a break, I'm gonna do today's New York Times connections puzzle. Let's see if I can do it. I don't want help from chat. I'm gonna hide that for now. Although, feel free to make fun of my bad guesses. Little Bulldog Sour Kiss. Husky Ring Stout. Gruff Pointer Horn. Amber Porter Horse Middle Fry. Well, yeah, you have whatever the obvious dog breed. Husky. I think Pointer is a dog. I don't know, that seems a little bit too obvious. Nothing's coming out as obvious. Stout is in small. Stout. Well, you have little, stout, husky, as in whatever. Large in size. My instinct. I am just gonna go for it. If this is like 1 away, not even close. Then is it dog breeds? A bulldog, a husky, a stout, and a pointer? Nope, not even close there either. Rough horse. Gruff and Fry. 1 away. Only have 1 guess left. Well, this is a particularly bad one for me. Rough horse. I don't know which one. It's the one that I guessed. I don't know. I got nothing. Definitely a fail on this one. Yeah, raspy voice. Little finger, okay. French bulldog, French fry, French horn, French kiss. Of course it becomes obvious. Well, better luck next time. That was a particularly bad one. You combat apologist, in doing so you have to know 10 times. Yeah, absolutely. And jobs. Well, I would say, yeah, don't sell yourself short. Definitely PhDs have their limitations. Absolutely. Okay, so what? Oh wow, welcome. Well, welcome, raiders. Thank you so much for the raid, TALORX. Super appreciated. Welcome, Energy Flow. Let's see if I can get my shoutouts working properly. Thank you so much for the raid, and always a pleasure to have you in class and do the lecture. Super grateful, as always. Always a great time, and well, now I'll be going over this paper that I very briefly covered in the lecture, or alluded to. No, you're absolutely right. People with PhDs can be arrogant. Absolutely. That has been my experience. So, yeah, we briefly, very briefly went over this paper, okay, during the lecture. And now we're going to do a little bit of a deeper dive. We're not going to dissect it, you know, from beginning to end. Maybe just go over the introduction, and, you know, go over maybe some of the figures, and then, you know, go over their main conclusions and findings, and give a little bit of more insight on like what they're doing, a little bit on how they're doing what they're doing. But even I myself, I cannot say with super detail what they're doing, because that's not my area of expertise. This is nature, ecology, and evolution, okay? And I am not an ecologist. And obviously, you know, I know quite a bit about evolution, but I'm absolutely not a world-renowned expert when it comes to evolution. And definitely not when it comes to ecology and evolution. So this paper is outside my area of expertise. But I overall know, like, the broad strokes of what they're doing. Yeah, exactly. There's a bunch of researchers here, like a dozen or so, from various institutions. The affiliations are probably towards the end there. And you obviously have your supplemental. Whoopsies. Don't know how to scroll properly. Where are the affiliations? Unless they're back here at the top. Oh, well, full list up here at the end of the paper. Yeah, at the end of the paper. Looks like University of Bristol. Acknowledgements, author contributions. Oh, here they are. It's wild that life would develop so soon after the formation. Yeah, absolutely. Well, yeah. Actually, even I can't say. In terms of early Earth, I don't know if there was water or not. I imagine there was some sort of water environment, but actually, even I don't know. This is like my bias and my limitations in my knowledge, where since I'm a biologist or biochemist, like, you know, and, you know, I'm taught that life started at 3.5 billion years ago, I don't really care what happens before that. It's like, I don't care about the starting of the Earth. Only, like, you know, the starting of life and the last universal common ancestor. So I don't look back before that of, like, what did Earth on day 0 look like? Was there water there? I assume so, but I don't actually know. No, some people think that way, and like I would want to dispel that myth. That absolutely, as a scientist, like, I don't know everything, and like what I do know, or, you know, if I know anything, like, it's mostly about stuff that most people don't care about. Or only a handful of people in the world care about. So we went over this a little bit, or at least the first part of the introduction, during, you know, the lecture. So the but we're going to now pretty much go over the full introduction, but even then, the full introduction is not super long. So yeah, the common ancestry of all extant cellular life is evidenced by the universal genetic code, machinery for protein synthesis, shared chirality of the almost universal set of 20 amino acids, and use of ATP as a common energy currency. The last universal common ancestor, Luca, is the node on the tree of life from which the fundamental prokaryotic domains, archaea and bacteria, diverge. As such, our understanding of Luca impacts our understanding of the early evolution of life on Earth. Was Luca a simple or complex organism? So this is what we, you know, briefly covered during the lecture. But then it goes on. And you're absolutely right, you know, no one can know everything. As much as I would like to know everything, and I try to get as much information as possible about things, you know, we all have limitations in our knowledge. Huge limitations in our knowledge, myself included. So what kind of environment did it inhabit, and when? Previous estimates of Luca are in conflict either due to conceptual disagreement about what Luca is, or as a result of different methodological approaches and data. Where, again, I'm not an expert in this field, but, you know, there's multiple levels of analysis when it comes to, you know, trying to figure out what Luca is or was. And, depending on which methodology researchers use, just as these researchers, they used a particular methodology, and they came up with like this answer that might be a better answer than what was currently. Although again, it's not like set in stone that these people are absolutely correct. But then, yeah, based off of maybe another group of researchers, their analysis, they might come up with, something different, or maybe even something better than this. So, where was I? Yeah, published analyses differ in their inferences of Luca's genome from conservative estimates of 80 orthologous proteins up to 1,500 different potential gene families. So yeah, these 80 orthologous proteins, you can just think of it as, 80, semi-related proteins. Fancy terminology for that, very roughly speaking. And like, yeah, had about, 1,500 different potential gene families, or, collection of different genes. Interpretations range from little beyond an information processing and metabolic core through to a prokaryote grade organism with much of the gene repertoire of modern archaea and bacteria. Or essentially kind of like saying of, what we see now in terms of prokaryotes today, like bacteria and archaea, maybe, or certain analysis leads us to believe that, the archaea and bacteria we see today pretty much resemble what was there during the last universal common ancestor, or the last universal common ancestor resembles what we see today in terms of bacteria and archaea. Or basically, in a nutshell, Luca appeared to be prokaryotic in nature, and that's effectively how we classify it now. Of the last universal common ancestor seemed to be a prokaryote, or some version of that. But recently reviewed in reference 7, here we use molecular clock methodology, horizontal gene transfer aware phylogenetic reconciliation, and existing biogeochemical models to address questions about Luca's age, gene content, metabolism, and impact on the early Earth system. So, even I cannot fully translate what, I only know a little bit of like what they mean when they say molecular clock methodology. I know what they mean when they say horizontal gene transfer, but I don't know what they mean when they say horizontal gene transfer aware phylogenetic reconciliation. That's outside my domain of knowledge, but they're doing obviously some type of fancy analysis here. Let's see. Water first shows up in our geological record about 200 million years after the formation of the Earth. It's found in ancient zircon crystals, which were found to contain traces of water, suggesting the presence of liquid water during this period 4.3 billion years ago. I will absolutely take your word for it, since it seems like you have done your research, and I can say confidently that I have not done my research. Or at least I'm sure I learned it in a class at some point, but that's like gone from my brain. So, estimating the age of Luca. So life's evolutionary timescale is typically calibrated to the oldest fossil occurrences, where again, we can only investigate, what we can find. As in, if there's no fossil record, it becomes virtually impossible, or like super challenging to, dive into the past. However, the veracity of fossil discoveries from the early Archean period has been contested. Relaxed Bayesian node-calibrated molecular clock approaches provide a means of integrating the sparse fossil and geochemical record of early life with the information provided by molecular data. However, constraining Luca's age is challenging due to limited prokaryote fossil calibrations and the uncertainty in their placement on the phylogeny. Molecular clock estimates of Luca have relied on conserved universal single copy marker genes within phylogenies for which Luca represented the root. So, yeah. Again, not my area of expertise. You know, I definitely have taken, you know, some microbiology and advanced microbiology courses, but like, you know, only a couple of courses at the undergrad level. And so nothing super advanced. But I only know a little bit of what they're doing here. Like, yeah, these molecular clock estimates of LUCA have relied on conserved universal single copy marker genes within phylogenies. Okay, so yeah, when they say like molecular clock, and specifically when they say, yeah, relaxed Bayesian node calibrated molecular clock approaches provide a means. Okay, I'll summarize as best I can. You know, ultimately, they're trying to look back in time. Okay, they're trying to look back in evolutionary time. And what we know is, like, through evolution, well, mutations happen, or changes in genes happen, okay? And so there's a little bit of a clock there, so to speak, as in we can kind of calculate, okay, or we can kind of backtrack of like, well, if there's this amount of change over time at a genetic level, and we know sort of the rate of genetic change over time, of like whatever, you know, 10 variations per X number of years. I'm totally making numbers up, but just for the sake of example, they can kind of like backtrack of like, well, if I know the rate of evolutionary change at a genetic level, I can kind of like start rewinding the clock. Because, you know, I'm sort of estimating the ballpark of like how the changes occur over time. And so with that, I can like start calibrating and start, you know, moving back the genetic clock, so to speak. But when they say this relaxed Bayesian node calibrated molecular clock, I believe, if I remember correctly, where, yeah, evolution, okay, occurs, you know, at a molecular level of, you know, changes in the genetic code. And so there's a change in the genetic code over time, okay? And, you know, there's a certain rate of change, as mentioned. Like, yeah, you know, every X number of years, there's going to be, you know, a change in the genetic code. But we know through evolution that, you know, when species diverge, okay, again, I'm just going by based off of my limited knowledge now of like what I remember from, you know, my microbiology. I could be getting this wrong. But, you know, well, certain species or certain groups of organisms or a collection of organisms, you know, they evolve faster, like, over time. Just, you can think of it as a generation time of like when it comes to the generation of humans, or generation 1 to generation of humans, generation 1 to generation 2 of humans, that's like on the time scale of like, you know, whatever, 50 years, 100 years, you know, a century. But if you're talking about, let's say, bacteria, okay, so like when you're looking at the changes, at the genetic level in humans, okay, the change is, you know, fairly slow if you're comparing it to, let's say, bacteria, or specifically E. coli, where E. coli, they have a generation time of 20 minutes, okay, where if every 20 minutes you have a new generation of E. coli, well, they can basically accumulate changes in their genetic code faster over time, as opposed to humans, okay? If you want 3 generations of humans, okay, then you need at least, you know, whatever, 50 years, 100 years, if not more. And so there's the rate of change in the genetic code of humans is much slower than, let's say, you know, 3 generations of E. coli, which will be like, you know, an hour and a half. And so this, as they say, this node calibrated molecular clock, I believe, I could totally be mistaken, but I believe it's taking that aspect into account of like, they don't just assume a consistent rate of change at the genetic level over evolutionary history. I believe they try to take into account of like certain populations accumulate changes in their genetic code faster than others, using the extreme examples of like, you know, E. coli and humans. where you can generate, dozens and dozens of generations of E. coli within hours or days, as opposed to if you want generations and generations of humans, you need like, centuries or like millennia. And so there will be less changes in our genetic code in comparison to organisms with a much shorter generation time. And so I believe, again, could be totally wrong, but I believe this sort of approach that they're talking about, this relaxed Bayesian node calibrated molecular clock, takes that into account. Well, let me see. Going back to what they mean on horizontal gene transfer, aware phylogeny. One approach is using software like AVP, alienness versus predictor. Yeah, I've heard of it, but don't know anything about it, which automates the detection of horizontal gene transfer by evaluating phylogenetic trees without relying on reference species tree, reducing human bias. Never let me said in biologists and have, biologists and geneticists do, open up any genetics textbook and look up the naming of genes, as I'm sure you know, and like the naming of genes can get, super out there, and wild and clever. So yeah, where was I? Almost, and yeah, as you sort of allude to, Jack, where rooting your phylogenetic tree, is super critical. And let's say not a trivial thing. Where, sort of like the phylogenetic tree that we're sort of referring to, sort of like looks a little bit like this. Like, yeah, things, branching off. Yeah, Sonic Hedgehog. You have genes named after Lord of the Rings characters, Beatrix Potter characters, etc., etc. So, where was I? Oh yeah, the root in these gene trees represents this duplication preceding Luca, whereas Luca is represented by 2 descendant nodes. Okay. Use of these universal paralogs also has the advantage that the same calibrations can be applied at least twice. After duplication, the same species divergences are represented on both sides of the gene tree and thus can be assumed to have the same age. Yes. They're going back. It's a bunch of jargon. I don't necessarily want to dissect right now. But we're going to get into some of the figures where, so I showed this briefly during the lecture. Just wanted to show it. I didn't want to go into detail, and we even now don't really want to go into detail because there's obviously a lot of stuff here. But here, okay, based off of the research in this paper, okay, where here they're showing pretty much just about everything we know about metabolism, or this very zoomed out version of pretty much all of metabolism that we know of like lipid metabolism, amino acid metabolism, blah blah blah, the different sort of subsections or categories of metabolism that we have mapped out as biochemists. I shouldn't say we, but other much smarter people. And so we have this giant network of metabolism that we know of. And so shown here, the important part is like the black lines, okay? So the entire figure, represents, loosely speaking, our entire knowledge of metabolism. And in black, it's like, well, those are the parts of metabolism that they think, or based off of their research, those are the metabolic pathways that Luca possesses based off of their analysis, okay? You can see sort of like in the categories that it branches out into. Where basically, based off of this figure, okay, since under the category of lipid metabolism, okay, therefore, Luca, based off of their analysis, did not have any sort of metabolism associated, or they could not undergo lipid metabolism, okay? But you can see there's this overlap with amino acid metabolism, which makes sense. And there's a little bit of overlap. You have some black lines under energy metabolism. Nucleotide metabolism, that's, fancy terminology for, we went over nucleotides during the lecture, so For anyone that's still around, you should at least know what nucleotides are now. Basically referring to, DNA metabolism, so to speak, loosely speaking. And like, yeah. And therefore, again, based off of this figure, glycan biosynthesis and metabolism, Luca did not have anything associated with that, based off of their analysis. Yeah, this is a super dense paper. That's why, like, I don't want to dissect it. No, well, one, don't want to dissect it because it would take forever, and two, because I'm definitely not qualified to dissect it in its totality. I can, like, extract the broad strokes, just like I did here in this figure, where, yeah, probabilistic estimates of metabolic networks from modern life that were present in Luca. In black, enzymes and metabolic pathways inferred to be present in Luca with at least pp, some sort of probability of 0.75. Or, some sort of statistical, whatever, analysis of 0.75. I can't remember what pp stands for. And then, yeah, shown here, again, I showed this during the lecture. This is their depiction of what Luca looked like. The first cell, and, this is what they hypothesize or theorize to be what's, inside there, what's going on there. You have your nucleotides, okay, your A, U, C, and G. So, based off of this, they're inferring that it was RNA-based and not DNA-based, but because you only find the nucleotide uracil in RNA. If it was DNA, this U would be a T. You still have a ribosome, okay, and that's part of translation. And it has ATP synthase for energy production. And, all the other stuff that we would sort of mostly expect of, yeah, amino acid biosynthesis. And DNA replication machinery, presumably. And here in the other figures, they're showing sort of, again, a little bit of, let's say, the ecosystem, or what they think the ecosystem that was surrounding Luca at the time. Where, again, it wasn't just, you didn't go from, like, absolutely nothing going on and then the first cell. There appeared to be some sort of build-up to the first cell. As in, before cellular life, there was some sort of precursor to that. And in fact, there's a term for that. It's like pro-something, progenotic, if I remember correctly. Something to that effect, because they use that term. Let's see. Oh, thank you. Probably deposit predict. Yeah, so some sort of statistical analysis saying, hey, this is how sure we are. Where is that? It is progenotic, I'm pretty sure. I'm just gonna search for it. Yeah. There it is. So yeah, the result is a picture of a cellular organism that was prokaryote grade rather than progenotic, and that probably existed as a component of an ecosystem, using the WLP for acetogenic growth and carbon fixation. Okay, so, like, yeah, they're essentially saying that, again, based off of their analysis, okay, you get Luca that looked like a prokaryotic organism. And not progenotic, okay, where this progenotic, I believe, is the term in ecology that's used for, like, again, there appeared to be something before the first cell, before Luca, but we don't really know what that looked like. Of, like, oh, thank you so much for the lurk, Gina Jake, super appreciated. Yeah, this progenotic, I believe, pretty much just means, like, before, genetic life, where Luca had genes. They even say explicitly that they think it had about 1500 genes or like 1800. We'll just go over their main conclusions. See if I can get to them. Or I guess the entire paper is, like, here's the conclusions, but there's no formal discussion section. Where... Yes. So yeah, all together, our metabolic reconstruction suggests that Luca was a relatively complex organism, similar to extant archaea and bacteria, on the basis of ancient duplications of the SEC and ATP synthase genes before Luca, along with high probability of positive prediction for key components of those systems, membrane-bound ATP synthase subunits, genes involved in peptidoglycan synthesis, and the cytoskeletal actin-like protein, MREB. It is highly likely that Luca possessed the core cellular apparatus of modern prokaryotic life. This might include the basic constituents of a phospholipid membrane, okay, since pretty much we consider cellular life as like, yeah, you need some sort of membrane, or a membrane composed of something specific, such as a phospholipid or some sort of lipid membrane. Although our analysis did not conclusively establish its composition. So yeah, it looked like some sort of phospholipid, but we don't know exactly the exact components of that phospholipid. In particular, we recovered the following enzymes involved in the synthesis of ether and ester lipids: alkyl dihydroxyacetone phosphate synthase, glycerol 3-phosphate and glycerol 1-phosphate, and components of the mevalonate pathway, blah, blah, blah. You know, again, a bunch of biochemistry jargon. Pathways, genes, proteins. The term progenetic has been used to describe, yeah, hypothetical primitive form of life that existed before, exactly, the divergence of prokaryotes and eukaryotes. Yeah, exactly. You need some sort of barrier. Well, yeah, you need something that, like, separates inside from outside, and that, you know, allows you to, you know, work against entropy, so to speak, or, you know, to exchange things with the surrounding environment. And that's why they say, you know, looks like, okay, Luca, whatever, evolved or came into existence into some sort of ecosystem already. And I think in here, yeah, in panel D, Luca within the context of an early ecosystem, the CO2 and H2 that fueled Luca's plausibly acetogenic metabolism could have come from both geochemical and biotic inputs. So they're kind of like saying of like, yeah, Luca, the stuff that Luca was using, the ingredients that it was like living off of, like could have come from like naturally occurring stuff that was lying around, geochemical, and like, yeah, there's stuff that maybe other life could have produced. And that's why they say the biotic inputs. That's just fancy terminology for, you know, whatever life, so to speak. The organic matter and acetate that Luca produced could have created a niche for other metabolisms, including ones that recycled CO2 and H2, as in modern sediments. And then, yeah, Luca in an Earth system context. Acetogenic Luca could have been a key part of both surface and deep chemoautotrophic ecosystems powered by H2, hydrogen gas. And yeah, this H2 aspect, okay, where, you know, as they allude to in this paper, and well, there's also other evidence that leads, I think, researchers in the field to hypothesize or theorize that Luca started at like a hydrothermal vent, okay, because it seems like this hydrogen gas, okay, H2, is important for, you know, Luca coming into existence, or basically it needed a source of H2, and so it begs the question, well, where is the H2 coming from? And that's why they think that at hydrothermal vents, since they are rich in H2, well then Luca, there's strong reasons to believe and evidence to believe that Luca started at a, you know, hydrothermal vent, because that would be a rich source of H2. But yeah, if methanogens were also present, hydrogen would also be released to the atmosphere, converted to H2 by photochemistry, and thus recycled back to the surface ecosystem, boosting its productivity. Yeah, so like if there was another source of H2, okay, then maybe, okay, Luca didn't have to come from a hydrothermal vent. And that's why they say here this hydrothermal H input, where, yeah, it's being released. But since at the hydrothermal H input, that's like the source, there's good reasons to believe, and, you know, other analyses point to, like, well, maybe Luca started, you know, deep in the ocean at like a hydrothermal vent, because that would be that rich source of H2. Well, even this, believe me, don't blame yourself. Like, this also goes over my head as well, because like I'm not a microbiologist, even though my microbiology like is well above average. But one, it's rusty since it's been a while since, you know, I've took those courses, and two, yeah, it's not strictly my field now. So I would say don't be so hard on yourself, because, well, this, I think no matter what, this is a complicated paper, unless you're directly in this field. Oh yeah, and I think this other nice finding, cool finding, is we found support for the presence of 19 class 1 CRISPR-Cas effector protein families in the genome of Luca, including types 1 and 3. So yeah, this CRISPR-Cas system, most people, when they hear CRISPR, they think of gene editing or genome editing, which is fair, because that's what we are trying to use it for now these days as human researchers, and also as, you know, gene therapy. But the CRISPR-Cas system, okay, its original function, like where we discovered it, is like in bacteria, and part of the bacterial immune system, their immune defenses against viruses. And that's why, going back into the figure here of, like, yeah, what they think Luca looked like as the first cell, you see that they have here this early Cas-based immune system, this CRISPR-Cas, and so their analysis leads them to believe that Luca had some primitive version of an immune system. And, well, as I just read, they, based off of their analysis, they found evidence that, well, they had some of the CRISPR-Cas system, you know, the whatever the class 1 and class 3 proteins, and so it might have some primitive version of, you know, whatever the current CRISPR-Cas system that bacteria has, and so that, well, Luca had the origins, or like the starting point of, you know, current bacteria's immune system that we now as humans have like extracted for our own purposes, or repurposed for our own purposes of genome editing and gene editing. Yeah, the dark oxygen, I will take a note of that, and I'll try to dig up some of the research and go over it. Because, yeah, I definitely have been reading some of the headlines and a little bit of the stuff around that. So that would be a good topic to cover next time. So, I would say wrapping up, yes. Let's say the last few paragraphs. On the basis of thermodynamic considerations, CH4, which is methane, and CO2, carbon dioxide, are expected to be the eventual metabolic end products of the resulting ecosystem, with a small fraction of the initial hydrogen consumption buried as organic matter. The resulting flux of methane to the atmosphere would fuel photochemical H2 regeneration and associated productivity in the surface ocean. So yeah, this H, again, based off of, again, in this field, surrounding research about Luca. Again, it seems like this source of H2 is critically important, okay? Where, like, Luca, or early life, needed some source of H2, hydrogen gas. And so that's why there's this focus on, like, trying to answer that question of, like, where would this hydrogen gas be coming from, okay? And so, yeah, if there were methanogens, okay, the metabolic end products of the resulting ecosystem, where, yeah, if it was already there, if there was already an ecosystem that was doing this processing of methane and CO2, well, that can also be a source of H2, where that's effectively what they're saying. Of the resulting flux of CH4 methane to the atmosphere would fuel photochemical H2 regeneration and associated productivity in the surface ocean. So, like, yeah, if this was going on, then, you know, maybe Luca didn't have to originate at a hydrothermal vent, because at the surface of the water, there would be enough of a source of H2, or would be H2 rich enough that it can support Luca. But if that wasn't there, then, like, yeah, we would go back to, like, well, I guess maybe Luca would have had to have started at a hydrothermal vent for a rich source of H2. Existing models suggest the resulting global H2 recycling system is highly effective, such that the supply flux of H2 to the surface could have exceeded the volcanic input of H2 to the atmosphere by at least an order of magnitude, in turn implying that the productivity of such a biosphere was boosted by a comparable factor. Photochemical recycling to carbon monoxide would also have supported a surface niche for organisms consuming carbon monoxide. In deep ocean habitats, there could be some localized recycling of electrons, but a quantitative loss of highly insoluble H2 and methane to the atmosphere and minimal return after photochemical conversion to methane, of methane to H2 means global recycling to depth would be minimal. Hence, the surface environment for Luca could have become dominant, albeit recycling of the resulting organic matter could be spread through ocean depth. So the global net primary productivity of an early chemo-autotrophic biosphere, including acetogenic Luca and methanogens, could have been of order 1 times 10 to the 12 to 7 times 10 to the 12 moles of carbon per year. So 3 orders of magnitude less than today. So this word, chemo-autotrophic, that essentially, auto, Latin for self. So, like, when it comes to microbiology, we tend to have some 2 broad classifications of autotrophs or heterotrophs. Auto meaning self, hetero meaning other, and troph basically means, you know, food, so to speak, or whatever, eating, or something to that effect. But basically, autotroph effectively means, like, you can produce your own food, as in plants. Plants are autotrophs. They harness sunlight, they harness energy from sunlight, and then carbon dioxide from the atmosphere, and then they're able to make their own food in the form of, you know, carbohydrates, glucose, sugars, etc., etc. As opposed to, us as humans and whatever animals, we are heterotrophs, as in we need to consume either plants or animals or both in order to meet our energy demands or our food demands, where we cannot produce our own food. We have to consume others. So, chemo-autotrophic just effectively means, can produce your own foods, and the chemo, I always forget, I believe chemo means, like, a specific source, of, like, carbon. Either way, not critically important. The important part is, like, auto versus heterotrophic. Luca, of an early chemo-autotrophic biosphere, including, so Luca would be a chemo-autotrophic organism, ability to produce its own food, which makes sense, as in, at least intuitively, it would make sense that, well, if you're a Luca, if you're the first cell, the last universal common ancestor, well, if you're a heterotroph and you're relying on consuming other organisms, or the byproducts of other organisms, and you're basically, like, organism number 1, well, you got a problem. What are you going to consume around you? So it sort of makes sense that, like, when you're the first cell, you're not really heavily relying on other stuff to be around for you to meet your food demands, because you're number 1. So it sort of makes intuitive sense, and it Seems like based off of their analysis, they're like, yeah, Luca was an autotroph, could produce its own food. Yes, oxidizing organic as a sort. Yes, that is definitely the more formal definition for the more technical official definition. So the nutrient supply, for example nitrogen, required to support such a biosphere would need to balance that lost in the burial flux of organic matter. Earth surface redox balance dictates that hydrogen lost to space and burial of electrons, hydrogen must together balance input of electrons, hydrogen. Considering contemporary H2 inputs and the above estimate of net primary productivity, this suggests a maximum burial flux in the order of 10 to the 12 moles of carbon per year, which with contemporary stoichiometry, blah, 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 could demand, blah, blah, blah. So all they're saying is, well, trying to get an insight into, the chemical makeup of, early Earth, or what was surrounding Luca, and how does that compare to today? Sort of the composition of nitrogen, carbon, hydrogen cyanide. Lightning would have provided a source of nitrate and nitrite and nitrate, consistent with Luca's inferred pathways of nitrite and possibly nitrate reduction. However, it would only have been of the order 3 times 10 to the 9 moles of nitrogen per year. So yeah, instead, in a global hydrogen recycling system, hydrogen cyanide from photochemistry higher in the atmosphere, deposited and hydrolyzed to ammonia in water, would have increased available nitrogen supply by orders of magnitude towards 3 times 10 to the 12 moles of nitrogen per year. So yeah, this aspect of ammonia, so, it's critically important of not only generally speaking, do, organisms need a source of carbon, okay? They also need a source of nitrogen, okay? And they don't just need any random nitrogen, as in there is nitrogen gas in the air in the form of N2, nitrogen gas, as they say somewhere here. Maybe they don't. Let's see. They do mention something about N2. But regardless, it's important. Okay, well, even today, that's actually, making NH3 ammonia is like super important when it comes to fertilizer and like plant production and agriculture. Where we call that nitrogen fixation, of like how do we basically get a usable form of nitrogen, as in the nitrogen that we find in air, which is nitrogen gas, N2, basically not usable for us, and so we need to find a way, or organisms need to find a way to convert nitrogen gas or make some sort of form of nitrogen that is usable, and ammonia, NH3, that is a usable form of nitrogen. And so the metabolic pathway of what we call nitrogen fixation is like super critical. And that's why they're analyzing nitrogen in this way. Let's see. Yeah, this hydrogen cyanide pathway is consistent with the anomalously light nitrogen isotopic composition of the earliest plausible biogenic matter of 3.8 to 3.7 gigaannum. Although that considerably postdates our inferred age of Luca, these considerations suggest that the proposed Luca biosphere would have been energy or hydrogen limited, not nitrogen limited. Yes, I would assume much easier, but I don't actually know for certain. But yeah, just as they say here, carbon fixation of basically fixing carbon. That's, effectively, in some sense, taking carbon dioxide from the air, or in the atmosphere and converting it into, carbon that's usable. Carbon with a bunch of hydrogen, as in CO2, no hydrogen. Sugar, carbohydrates, well, that's carbon with hydrogen. And well, plants do that. Plants, that's why we as humans, we are dependent on plants. Without plants, we would not be here. Where we rely on them, not only to, consume the carbon dioxide that we exhale, but also provide us with oxygen, okay, as well. And also they're like a major source of carbon fixation, of like taking carbon dioxide from the atmosphere, okay, and converting it into something that's usable even for us, okay. Sugars, carbohydrates, glucose, okay. Well, we eat plant matter, we eat fructose. And so just as you have carbon fixation, taking an unusable form of carbon and getting it to a usable form to us, you know, we need the same process when it comes to nitrogen, as in nitrogen fixation. Taking an unusable form of nitrogen in the atmosphere and converting it into something that's usable for us. And even we as humans, okay, well, we can do it artificially, called the Haber-Bosch process, but we do it way less efficiently than microorganisms. Where the Haber-Bosch process is like a super energy-intensive process. It's super important, okay, but we just cannot compete with the ability of some organisms to just take unusable nitrogen and turn it into ammonia. We have to expend a lot of energy, a lot of heat. That like is actually kind of like a huge problem. Yeah, exactly. Can be created from methane, H2, and yeah, this N2. I appreciate the subscript, and that's why they refer to this lightning earlier or multiple times. Yeah. Lightning would have provided a source of nitrate and nitrite and nitrate. Well, I always get nitrite and nitrate mixed up. Where yeah, I believe nitrite is NO2 minus and nitrate is NO3 minus. I can never get those in my brain. Even now, I better double check. Yeah. So yeah, nitrite, NO2 minus, nitrate, NO3 minus. Yes. But I always get them mixed up. I mean, it's always good to, you know, fact check yourself. I'm always sort of like doubting myself. Always trying, or at least do my best to make sure that, you know, I get things accurate to the best of my knowledge. And even now, like, even when, like, I'm covering stuff, like, in my own field of, like, the biochemistry stuff, you know, I consistently, like, second guess myself. Or I'm always trying to make sure that, you know, I'm being, you know, as accurate and hyper-specific as possible. So yeah, wrapping up, okay, going over the conclusions again. Just want to pretty much go through the entire conclusion. We just read the last part of it during the lecture. So yeah, what do they find? Conclusions. By treating gene presence probabilistically, our reconstruction maps many more genes, about 2,600. So yeah, so based off of their research, they find that, you know, they point to, you know, Luca having 2,600 genes. Let's say for reference, you know, humans, us as humans, well, depending on which textbook, you know, we have 18,000 to 20,000 genes, okay? So Luca has about, you know, 10%, 8% as many genes as us. So yeah, our reconstruction maps many more genes, 2,600 or 2,657 to Luca, than previous analyses and results in an estimate of Luca's genome size, 2.75 megabases. That is within the range of modern prokaryotes. The result is a picture of a cellular organism that was prokaryote grade rather than pre-genotic, and that probably existed as a component of an ecosystem, using the WLP for acetogenic growth and carbon fixation, We cannot use phylogenetics to reconstruct other members of this early ecosystem, but we can infer their physiologies based on the metabolic inputs and outputs of LUCA. How evolution proceeded from the origin of life to early communities at the time of LUCA remains an open question, but the inferred age of LUCA, 4.2 gigaannum, so that's, fancy terminology for 4.2 billion years ago, giga, billion, or gigaannum, translates to billion years, compared with the origin of the Earth and Moon, suggests that the process required a surprisingly short interval of geologic time. Where, yeah, pretty much if their estimates are correct, and, they are not saying, and, the field is not saying, like, let's revise all the textbooks, but if in fact they are, then it's like, yeah, it took maybe approximately 200, 300 million years before, like, the formation of Earth, and then the first last universal common ancestor, or the first cell, basically. Yeah, we should always be skeptical, especially of our own, absolutely. And, well, not to, I guess, be morbid or negative, but this is just a reality of, like, well, yeah, my grandmother, who's 91, okay, who's, in the hospital recovering from a fall. Her recovery's going well, but yeah, the cognitive faculties and the memory is just like, it's steeply declining, alarmingly, and sadly so, but that's the reality. And that sort of, like, makes me question my memory even more, because, like, if you ask her, okay, like, she's gonna be dead certain about something, okay, which, who am I to, question her, so to speak, because, like, that's what she has in her brain, like, and she has no control over what she remembers or what she doesn't remember. And so it's, it's one of the reasons why I'm even more skeptical today, just because, our memories are super flawed, and that becomes crystal clear when, well, you encounter people, or you encounter the reality of cognitive decline, and, like, if you can't remember just, like, even 5 minutes ago, it's super alarming. Which would be some minor retweaking, but everything else, yeah, absolutely. No, it is not fun, but it is the reality. At the end of the day, I love my grandmother. I'm super grateful that she's still around. Okay, she's 91. Okay, so already she's, like, beaten the odds of life expectancy. But, like, yeah, she's living 2 minutes at a time, or 5 minutes at a time at best, depending on what the memory is. Okay, if it's a memory about a song that she knows, okay, then that's like etched in stone, or, like, that's still clearly intact. Of, like, a song that she knows from her childhood, she'll know the words backwards and forwards. But, like, yeah, like, what's for breakfast, the fresh information. So, like, basically, okay, all of the long, not all, but a significant portion of the long-term memories that are, like, etched in stone, so to speak, are still intact. But, like, yeah, acquiring new memories, like, I think is mostly, or essentially out of the question now. And, like, yeah, even, like, those memories etched in stone are slowly being withered away, unfortunately, but, like, that's the reality. I'm not a neuroscientist, but I know the realities of the brain, and the brain changes that occur over time with aging. Just as I'm sure if you scan my brain today at my age and compare it to how I was 20 years ago, like, there will be clear differences. And, at some point, cognitive decline comes for us all. Yeah, absolutely. Now, like, the immediate goal is, hopefully, optimistically speaking, is getting my grandmother at least back on her feet, at least with a walker. Okay, that would be a huge milestone. That would be, like, pretty much the ideal outcome. Before the fall, she was able to essentially walk unassisted. Now, the realistic goal is to get her to walk assisted. And when it comes to the recovery of her legs, it's going actually pretty surprisingly well given her age. And yeah, it could have been much worse. Even though it's not the funnest right now, but yeah, at the end of the day, I'm not religious, but every day is a blessing at this point. And we're overall just lucky that she's still around and overall decently functional. And it's all about, keeping the good times, trying to make her as happy and as comfortable as possible, to the best of our ability. That's what it's about. That's the reality. And thank you so much for the kind words, Jack. Super appreciated. So that's pretty much all I want to cover for today. Well, covered quite a bit, the lecture. And yeah, going over the paper. I'm going to, try and always... well, I'm not going to make any guarantees or promises. But for each lecture, I'm going to try and, pull in a paper that's relevant. And maybe go over the paper in more detail after the lecture. But as you mentioned, Jack, I will probably, for next time, go over that dark oxygen after next week's lecture. That's a great idea. And I should absolutely be able to relate it to biochemistry. That shouldn't be a problem. So that's all I want to cover for today. I'm still getting back into the swing of things of regular streaming. As mentioned, we missed last week, just a slight misunderstanding. But we should be back to our regular schedule, weekly, for the Wednesday lecture. And I'm hoping, no guarantees, but I'm hoping this Sunday is when I get back to doing my regular content of covering research, going over papers like these, but papers mostly in my field that I'm way more comfortable in dissecting, or just other research that I find interesting. But yeah, I think this Sunday I should also be streaming again, or getting back to my regular schedule of streaming on Sundays, and hopefully moving forward, Wednesdays and Sundays. Well, Wednesdays should be definitely for sure, since I'm committed now to doing the lectures. Then also hopefully Sunday as well. But that's all I prepared for today. Let's see if I can find someone to raid. Moo Hoodles is streaming. Yeah, that's a fantastic idea. I would love to have, if ANCOM wants to have some sort of a research roundtable or, discuss anything, absolutely, with just about any scientist, I'm always game. Well, yeah, no, I'm trying to strike the balance, even though, I guess I'm a little bit selfish. I mostly care about covering the content that I want to cover, but I do take audience suggestions. I recognize I'm not the most exciting streamer on the planet, nor do I desire to be. But I'm trying to strike some sort of balance between information and somewhat mild entertainment. I think we're going to raid into Moo Hoodles. On the topic of Luca and the last universal common ancestor, she covers astrobiology, so I think that's the perfect streamer to raid into. All right, gonna set that up. But yeah, if you're not following Moo Hoodles, you definitely should be, because she is a science and tech streamer. One of the original, if not arguably one of the founders of streaming science and technology. I tend to pretty much follow that. That's my guiding principle of lowest common denominator. And at the end of the day, I'm grateful for the audience that I've cultivated, that want to engage, that want to learn. that want to challenge, okay, that want to call me out when I make mistakes, which I absolutely do, and it's bound to happen over and over again. But yeah, we're going to right into Muhudos. Thank you everyone for passing by. Thank you, Jack, for passing by and all the wonderful, information that you provided, and the suggestions and the feedback. And thank you, Bob Marley. Thank you, Orange Hue. Thank you, TALORX, for the raid. Thank you, Energy Flow, for passing by. So I should be back this Sunday covering getting back to science, research, and news. If not, okay, Wednesday should be the usual scheduled lecture number 5, getting back into diving deeper into proteins. But yeah, that's it for now. Follow Mahoodles, and until next time, take care and be well.